Recording in progress.
Halo semuanya, Assalamualaikum. Salam.
Good morning.
Good morning, Mr. Michal.
Good morning, Professor Tresna Lestari.
I'm not professor.
Can you hear me?
You
Okay.
can call me just by my name.
Okay, Tresna, hello.
Hello.
Can you hear me?
Yeah, I can hear you clearly. What
Is it okay?
the other guys? Can you hear Mr. Michal voice?
Yes, I can hear. I hope the voice will be clear.
Yeah.
Because if I open camera and speak, maybe sometimes the internet.
Oh, yeah.
Internet may be not sufficient.
Yeah.
So if you cannot hear, please let me know if there is any problem with the connection.
Yeah.
So I can, maybe I will turn off the video or do some change.
Yeah. Okay. Okay, are you ready, Mr. Michal, to start our agenda today?
Line. Is everything okay? Yes.
Kayaknya ini ya.
Okay. So we can start already.
Audiensnya gimana? Suaranya kedengaran nggak ya? Suaranya jelas nggak?
Ada Ibu.
Jelas Bu.
Ada jelas.
Jelas Bu.
Di saya aja sinyalnya, takutnya ini di saya agak putus-putus sinyalnya. Oke, mungkin bisa kita mulai saja ya. Good morning everyone, and good morning Mr. Michal. I'm happy to have you here this morning to give us your lecture. Actually, this program is a collaboration between two universities. So we have the student from Bakti Tunas Husada University and Buana Perjuangan, Universitas Buana Perjuangan from Karawang. Actually, from Universitas Buana Perjuangan Karawang, we have Dr. Haji Puji Isyanto, SEMM. He wants to make a brief introduction with Mr. Michal, but I think he is not in the Zoom room yet, but maybe later after the presentation, he will join us in the room Zoom. So he can contact you or give you a brief introduction. I think we can start the material. Mr. Michal, you can start your lecture now. Your time is yours. The time is yours now.
Okay. Thank you very much, Tresna, for kind introduction and also for kind invitation for today's, I would say, a session, the research session. So, hello everybody. It is my pleasure to meet you. Senang bertemu denganmu. And I will, in my slides, I will slowly, basically my journey through the studies from the Farmasi bachelor, master, Ph.D. to be a researcher. So let me. Okay. So In our study, wait a second, I think I will put this one down here. Yeah. Okay. So in these maybe 30 minutes or 1 hour, we will have discussion on the bioactive nature products that basically I study for a long time. And I particularly will also introduce some of the bioinformatic tools. So I am from Kaohsiung Medical University from Taiwan, and our institute is Graduate Institute of Nature Product. We belong to College of Pharmacy. Our university has medicine and also life sciences, environmental sciences, chemistry, and these related majors. So it's more focused on the medical point of view. If anybody using LAT to contact me, and okay. I'm from Czech Republic. So Czech Republic is quite far from here in the heart of Europe. Our history is pretty interesting. You may know maybe this force that established the Charles University in 1348, one of the eldest universities in Europe. And also we had many monarchies and many kings that were leading our country together with Austrian Empire, because we were always part of the Austrian or together with the Romans and Austrians in the history, but actually we had our own culture and story. And the most recent history is the Czechoslovakia, that is the republic, together with Slovakia, was established in 1918, but in 1939 we were annexed by Hitler. And then was the Second World War, and after that communists from Soviet Union came to control our country for 40 years until 1989 when we did. But unfortunately we split with Slovaks due to some economic reasons, but we are very like brother countries with Czech and Slovak. And we entered NATO and EU. And for Czech Republic, if you know some Czech product, you may see the Škoda car. This is the old version, this is the new version of the same Škoda Octavia. We have beautiful glass, Swarovski. Our tradition is to make glass products. Nowadays, a little bit decreased because of the high and very competitive products from China. And also nanomaterials, and we like beer because beer is usually cheaper than water. If you go to pub, beer is cheaper than water. So that's why, and we have so many types of beers. So this is like Czech nature. And we had one Nobel Prize in 1959 for polygorography. So in research, we have still many things to do. Okay, this is my hometown. And you can see the city hall, the church. It's called Jablonec, that means apple town. So we have 17 apples here on the tree. This is our sign of this city. It's like 40,000 people city. It's not big. And actually I didn't show the map, but it's approximately 10,000 kilometers from Taiwan. So it's just in the middle next to Germany, Poland, and Austria. This is the university in Prague where I graduated from my pharmacy magister degree. We call it magister. And then on Pharm.Dr is additional degree. Actually, we have a little different system, but it's maybe you may think about it, it's equal to yours pharmacy degree. So this is Prague, the Charles Bridge. There are some nice views. This is the very, very old Orloi clock. It's like a sun clock, especially with some kind of puppets. And this is our faculty, which is actually in another city. It's a faculty of pharmacy since 1950s. And this is one of the towns that is protected by UNESCO. And here I will want to introduce myself. So after I graduated from pharmacy during my studies in Czech Republic, after that, actually I should say that one of the reasons that I went abroad is that I had always the nature like to explore. So in my second year, I went to US for the lifeguard summer job. In the third, fourth year, I went to Taiwan as a student exchange. And for 6 weeks, and I went to the community pharmacy. I also visited the lab of Professor Zhang for 1 day at KMU. And then in fourth year, I still went to Finland for half year for Erasmus program. This is European program for research exchange. Eventually, after 2 years, I went to Finland maybe in 2007. I also finished a degree that is a special one for Czech. It's Pharm.Dr. It's not totally equal to the Pharm.D in US. And just like I say, it's a little special. It's like additional research work for 1 year. And you can still work in pharmacy with this. You will get a little higher salary. But after working in pharmacy for 1 year, then I decided to contact Taiwanese Professor Chan and to try to see if there is opportunity to study. Maybe one of the reasons I choose Taiwan is also during my exchange, I met my current girlfriend. I studied PhD in the same institute that I'm now, so Graduate Institute of Natural Products. And then I went for postdoc, and I also was working with Taiwan European Union Innovation Cooperation Platform to link Taiwanese researchers with European researchers in NCKU, that is University in Tainan, for 1 year until I came to my current position. So the research area is the natural product chemistry, but I also did biology direction, and we studied traditional folk medicines mainly, and also we can use bioinformatic tools. And for my hobbies, I like some green technologies. I like to play football, but recently maybe less, or table tennis, biking. And since I have 2 boys, so I'm pretty much fighting with them playing. Yes. So let's go to the research. I would like to briefly introduce my topic I did in master, because that may be more close to you, that you may imagine your thesis topic and what kind of research. So basically I was working in the organic and inorganic chemistry department. Later the dean of the faculty, Alexander Hrabalek, and the young faculty member, Kateřina Vavrova. So we were synthesizing piperidine derivatives. Basically, we protected the group, and then we did esterification here. So we synthesized different derivatives of piperidine carboxylic acid with different chain. So it contained the polar head and non-polar chain. And then what was the interesting part that we tested this, the bioassay was actually on the real pig skin. And we cut this pig skin and put it here in the front cell. And then you apply your drug. Okay, I forgot to introduce. This is permeation assay. It means transdermal permeation. Transdermal permeation is we want to see how much of our drug can pass through this barrier. In this case, is the real skin, because we want to apply the drugs through the skin. So we, for example, dexamethasone, if it doesn't go through the skin well, we can apply enhancer and we can enhance the amount of the drug that pass together with our accelerant. So we will synthesize the accelerant. Then by HPLC, we can quantify the amount of the drug that passed through. And here, some of the results, so we synthesized different piperidine derivatives, like this dimer salt was very interesting. We had positive control that is based on open version of this piperidine cycle, and it was active. So some of our compounds effective, the C10, carbon 10 chain possessing compounds. And this was basically the scope of the work. Later on, during the doctoral pharmacy thesis, during the Erasmus in Finland, my supervisor, Karika Tikanen Kaukanen, and in Czech is Martin Doležal. So here the topic was a little bit different, but again, biological part, chemical part. We were studying the interaction between bacterium and host cell because in the interaction, the glycoprotein, the pili, some nature. May you hear me well? Right. So here we can see if our drug can inhibit this interaction, basically. And what drug we use here was a milk whey. That's the side product of cheese production, milk whey. Somebody use it for the protein to exercise, high protein intake. And in this study, we try to get sialic acid type oligosaccharides. So oligosaccharides means it contains sugars, maybe glucose units in several, maybe 3 to 7 only, not too many, not too few. And it contains sialic acid. We focus on that. We use detection to one method to detect the acidic ones versus neutral ones. So we focused on oligosaccharides because we think the oligosaccharides can inhibit this interaction. And the results, although the SD values are very big, show that some of these acidic ones are active. And we used different methods from the milk whey, from the nature product. We used Cefadex to separate the different sizes of sugars to get oligosaccharides fraction. And this is the principle of the ELISA assay used. So we isolated from a Neisseria meningitis that causes meningitis disease, the brain inflammation. So we isolated pili. The pili will usually bind to the receptor of the host cell and then infect the cell. So here, if we can block the pili by our oligosaccharide or nature product, then they have no way to bind here to the host cell glycoproteins. And this assay can basically visualize it based on the fluorescence. So this also I learned during my stay in Finland about the later on, when I joined the PhD, until this, that we take typhonium diacids inside for anti-allergic activities. Then we had saffron collaboration with Ukraine or Iris. Then with Professor Zhonglong Huang from Chang'an University, steroids and different volatile compounds from very popular for I ran anti-allergic essay and related molecular biology as well during my PhD studies. And later on, I will speak about the bioinformatics part, which is the molecular docking, like you can see here, or ChemGPS positioning in the chemical space that I will introduce later. So for my PhD dissertation, you can see that anti-allergic screening is inside and chemical and biological investigation on typhonium species, but also included the seeds of Aquila malacensis study. So this is basically overview, and here I will speak briefly about the allergy, why I chose it as a topic for my PhD and interested in this topic. And then some of the results on each of these topics. And in the end, I will talk about the molecular modeling and bioinformatics. So in allergy, you know that suffer from allergies also due to very, very clean environment nowadays. And that may be one of the main points why the allergies are getting more, because you can see the red parts are like the asthma and rhinitis are very much increasing in numbers nowadays, especially in the modern countries. So it's almost 20% population that will suffer from some kind of allergy. And myself, I actually also from childhood suffer from atopic dermatitis. And these all motivate me to study the professor who is studying this essay that I will introduce. And like this it's not my son, but when he can be dirty, he will be dirty like that. So I think this is a good way to strengthen your immune system and have less allergies. In the clinical field, actually, if you search for anti-allergic drugs for last 20 years, there is no much improvement because we still eat Zyrtec and these histamine inhibitors. It's interesting to see that gamma-linolenic acid, which is like fatty acid unsaturated, was registered and actually later withdrawn from the market, but it had proved some activity, but it was withdrawn due to some unstable results. But still, it's interesting to see this gamma-linolenic acid and some of its crude extract to be used as anti-allergic agent. And also, you can see that some grasses from some plants are used for allergic rhinitis as a drug, as maybe so-called new botanical drugs, or some pollen extracts are used for hypoimmunization. So there are still many natural products that are potential for development as anti-allergic drugs, although sometimes we just want to synthesize your body to prepare for that allergen. So that is a little different approach. Here we are trying to inhibit the degranulation, inhibit the release of histamine. And we detect it using one assay that is called beta-hexosaminidase, because this enzyme is released from the cell together with histamine. And in my course, I explored many extracts and fractions and compounds from our classmates, because Professor Chang's lab used to have 20 PhD students at one time in 2005 to 2010. So there were many topics, and I usually take a crude extract for the screening. And my topic then, I did it more on molecular biology, that is dealing with calcium and so on, FC receptor, for Ig receptor, etc. So, sorry. Okay, here we have the example of the screening. So basically, we use RBL cells and we find whether it's non-toxic. If it's non-toxic to the cell, we proceed to the assay, and we have chemical inducer and antigen inducer. We use these two inducers due to calcium one is more stable assay, antigen one is more human-like physiological related assay because it's like real antigen, IgE, dealing with Ig receptor. So these two assays I basically bring through my PhD studies to get the active components that are dealing with allergy. In this, or I would say, in the one of the main findings that we made is that Aquila malacensis seeds. This is a traditional medicine. Aquila malacensis is a tree. I didn't put the introduction here, but this is a tree that which bark is used for fragrance, for the religious and also medicinal reasons, and it is when you cut the bark, the resin will come, and the resin is very, very expensive, very, very expensive. And in Taiwan, in the east, we have many farms that are doing this business, or they are planting this tree. So we receive the seeds that they will basically throw away, and we extracted them, but there is a lot of oil. And later I will show you how we try to solve this problem that is seeds. So in the end, we identified the formable ester that is having the fatty acid linked here. So yes, we also isolated different derivatives. You can see also diglycerides. And we also studied together with Professor Huang. Vital is my classmate from India. So we work of the compounds had enhancing activity like formable PMA. You may know PMA. Some of the researchers may use it to induce inflammation. And these are basically derivatives of PMA because they have different side chain at different positions. And the side chain itself, like this compound one, was a new compound because it is aldehyde. And it had enhancing activity on superoxide anion in human neutrophils, but inhibitory activity on elastase release, which is very interesting feature to have enhancing in one and inhibitory in the second assay, because this elastase is also degranulated, released from the granules of neutrophils. We also studied the allergy. Oh, here is the introduction. Yes, sorry. This is used Agarwood. This is to show you the picture of the tree, of the bark, and the resin basically is coming here, and the seeds are like a side product. And basically, seeds has very scarce literature. That's why we also study it. And this is the story how we resolve the chemistry part, because it is seed, so it has a lot of oil, of course. So after traditional partition, we didn't work on hexane layer. We work after water partition. We work with methanol layer, and then we use different column silica, Cephadex, silica to obtain this compound. So plus the other compounds as well. So in here, we try to use the bioactivity guided fractionation to trace the bioactivity from the extract, because we found that during the screening, I found that this crude extract, ethanolic extract of the seeds, has very good activity with therapeutic index over 100. What is therapeutic index? Therapeutic index is the activity versus toxicity. So if this sample was non-toxic and this sample had IC50 above 1, so the therapeutic index will be 100 divided 1, so it's over 100. So you can see that the subfractions had even higher therapeutic index, up to 1 million therapeutic index. That means that our extract was active at nanomolar, even like up to picomolar level. So this is, or nanogram per ml in this case, because they were still fractions. This is very, very strong activity in the model that we used. But I need to know that I used 20 hours treatment. And because this is furball ester-like component containing sample, we assume that the furball ester may have some short-term enhancing effect, some deteriorating effect on the cell, although it's not killing the cell. So the result is very interesting, but as we know, the furball esters are enhancing for inflammation. This needs to be further studied. So there is still a room to explore more in this field. From the chemical point of view, this is a nice example how we do the NMR guided, not only and separation. So luckily in this case, the bioactivity was actually following this the same way. So that's why we choose this fraction based on bioactivity. But you can see that the peak, and you will not be able to notice this during the early stage of the research, only after you get all the data together. You can see that this peak is growing up, about 7.6 in NMR. And the 7.6 is a typical proton 1 signal in the structure. So this is like you have some different compounds have typical signals for different moieties or different structural features, like flavonoids or terpenoids and so on. So this is very typical for furball ester, together with this signal about 5.6. But there are many signals, so this signal is very typical. So based on this, we can trace the compound from the ethanol extract to methanol partition layer, fraction 4, and then subfraction 4,4. Of course, we need to perform structural elucidation for the compound 1, and we need to get all the NMR data to be able to connect this proton correlation to this carbon by BC. So we make sure that the linkage is here and not here or here. So definitely it's important to have the data to be able to do structural elucidation. And also the typical signal here is 7.6, you can see in NMR. So if you identify one type of target compounds with typical signals, then you can trace it through the NMR, basically. And for the long chain, because this was a new compound, it can be then identified based on the literature comparison. And I also wanted to speak more about bioinformatics, so I will go a little faster through the biological part, just to show you the overview which kind of activities, which kind of tests, which kind of assays we perform also in our institute currently, of course, will be by other professors that are dealing with pharmacology. Similar kind of assays they will also perform. So for example, this is IgE receptor expression using flow cytometry. Then this is interleukin-4 MCP1 mRNA PCR experiment. So here we choose the marker for interleukin-4, which is very strong in allergy because it's Th2 driven. Here, finally, we found that calcium influx. This is fluorescent microscope. This is online, live taking, and then you analyze the movie after every 5 seconds, and you have 1 point. So we can see the decrease in calcium influx into the cell after the treatment with our crude extract. And then correlated with calcium signaling pathway, PLC gamma 2 Western blotting we performed to identify these. For the chemistry part, because we always want to analyze the chemicals in the crude extract, and because that is very obvious from the NMR, there are typical signals for the fatty acids and also the double bond unsaturated ones. Interestingly, you can see these two signals, 0.8 and 0.9. So we can identify that, okay, I will go to this omega-6. Omega-6 will be 0.9 because the double bond here is more far away from the terminal metal group. But in omega-3, like we eat as a healthy food or the fish oil, the double bond is more close to the terminal metal group. So there is a shift in NMR to the left. So it will go 1 ppm for this terminal metal due to the double bond. It's just at 3 position. Here is at 6 position. So we can some of steroids. Plant contain a lot of steroids that I also isolated before, and a lot of fatty acids. There are not much flavonoids or the didn't. Acids we need to do acidation. We need to it will not be separate. It's only part of the real components. Actually, you may see only palmitic acid, and the one with double bonds may not be seen. So here we compare the two different species because this plant is used as folk medicine for cancer. I didn't introduce the plant itself, sorry for that. But that was the reason why we try to use this, because it's used by many folk people to treat inflammation and cancer. Yes, so this is the composition, mostly palmitic acid and linoleic acid. So no double bond and 2 double bonds components. So we compare them. And further, we have taken the isolated fatty acid because I did isolation on this material. And these isolated fatty acid contain hydroxy group in their structure, which is quite rare to be reported. And also we identified one of the new derivatives with a relative structure. We didn't have absolute stereochemistry here. So the point is that they were active in anti-allergic assay. And then we use NP to correlate these fatty acids in the chemical space. And we correlate these fatty acids, the circled ones are the most active, with commercial anti-allergic drugs. So here you can see my supervisor, Professor Chang and Professor Wu, doing my PhD course. And I will speak about the bioinformatics later on. The isolation scheme is like this. So we isolated these compounds. The structures are here. They are fatty acid with 2 double bonds and hydroxy group. The GC mass was not necessary to confirm the position of double bond and hydroxy group. Without GC mass, we are not going to success to identify these compounds because they are long chain compounds. And then in further study, I have purchased and obtained series of fatty acids without double bond, with double bond, 2 double bonds, including EPA, DHA, these omega-3 very famous fatty acids, hydroxylated fatty acids, our isolated fatty acids, and hydroxylated unsaturated fatty acids, like ricinoleic acid. It is used for some digestive problems. So this panel of fatty acids, we, except of ChemGPS, that will be the last step basically, we did bioactivity screening to find the structure-activity relationship. So in medicinal chemistry, you always want to get the best derivative structure-activity relationship. So we can identify that in inflammation, the double bond and free fatty acid, this is by Professor Huang. This preliminary study, we knew actually this already before, but here we additionally wanted to add the hydroxylated one. So the hydroxylated or non-hydroxylated all had very strong inhibitory activity in superoxide anion and elastase release assay. Simple oleic acid that you have in oil, in olive oil neutrophils, they are isolated from the human blood in this assay. So any oil, oleic acid, linoleic acid, double bond, unsaturated fatty acid in free form, they have strong inhibitory activity on human neutrophil function. This is one of the important message for everyone that you can take home today. And in here, the hydroxylated had the same activity level, the ones with double bonds. In allergy assay that was performed by me, you can see almost no activity here. And the level of the active ones is a really high concentration. So it's only hydroxylated fatty acid that was able to inhibit mast cells from the granulation. So the hydroxy was required for the activity, and together with double bond was better than without double bond. So then we put these into the ChemGPS basically, because I put that slides later on. So these compounds, the panel of the compound that you just have seen is in this cluster, and I will talk about that later. Okay, during my PhD, I tested a lot of samples, but it is interesting to say that although I tested many samples from my classmates in the lab, it has been only the collaborative Egypt samples that we kind of were able to finish the study, because if I tested the samples from my classmates, they always want to focus on their own studies. So in the end, the allergy part was not so important for them. That's why I tend to test more and more in my later later course for collaborate with the international scholars, because Dr. Shazli was our postdoc before in our lab, and we have good connection here. So actually we have quite many publications with the collaborators from abroad. One of them is, and here we also applied the docking. One of them is flavonoid, and by flavonoid from that is bicolor, the Iridacea plant. And here, although this is not a new compound, but one of the flavones was, we could correlate the elastase activity in human neutrophils and elastase enzyme to the compound. We could interact, they can dock together, they can interact well. So this was a good result. Also, we had fennel and cumin in Mediterranean diet contain a lot of these. I know that Indonesia, you also like spices so much. So here we did volatile oils analysis. Chisimas and Professor Huang did anti-inflammatory study with different receptors, FPR, and different pathway. This is basically all done by Professor Huang. And also Ms. Chen did antimicrobial activity in this study. So we have like collaboration. Olga Mikalenko from Ukraine, Kharkiv, also Farmasi. They study Iridacea, and we tested in all of the assays, including allergy inflammation virus, because later on we collaborated with antivirus assays and professors. So these are the two different studies. One is on saffron, Crocus sativus. Saffron, you may know saffron, is also expensive spice. So we study different parts of saffron, the inhibitory activity, and this is influenza virus and enterovirus. And the Iris hungarica, the bioactivity is related to antioxidant, but also anti-allergic, anti-inflammatory activity was observed. And the identification of compounds is based on UPLC mass together with the standards in this study. In another study, their collaborator from Lithuania, and we do basically bioactivity, and this is on saffron stigmas, because we, in previous study, it was different part of saffron. So saffron stigmas, the spice itself, but this is related more to cancer, this study. When I was postdoc in Professor Huang's lab, because Professor Huang is expert in neutrophils, some of the class, and actually TCM doctors, most of them are TCM doctors. So we have composed a review article on psoriasis, the effect of neutrophils in psoriasis, but also the ARDS related to COVID, treatments that may affect neutrophil function for treating the late stage complications with COVID that were so lethal during the COVID, especially during the COVID outbreak. So this is it's a really pleasure to work with Professor Huang. I want to thanks for him for his support. I would like to focus here on the molecular docking because I know that you are interested in the computer-based research. So I will have three topics. One is molecular docking, one is GPS, and one is GNPS. In molecular docking, to introduce in molecular docking. We want to predict the molecule interacts with the protein. So we choose small molecule. Okay, we can choose DNA also. And protein, some receptor, actually it can be also DNA. It can be DNA to DNA, but we do always molecule to protein because small molecule is even better. Otherwise it's getting too complicated. So we observe the hydrogen bonding, and basically the lower energy state, the more stable, the lower the better. So these are the two features that we want to observe. And there are many softwares. Myself, I learned how to do Autodoc. There are different like Discovery Studio. Also, there are online engines. You can enter the drug into some of the engines and they will calculate the energy. But I think the fundamental question is how to design the study correctly. And you can see there are many docking studies nowadays, a lot of bioinformatics, how to know which study is good, what is the good design. So there are two ways. And one of them is to dock libraries of compounds, thousands and millions of compounds, and choose one with the best energy, best hydrogen bonding, because these are important for the interaction and inhibition of some of the protein. So do the docking first and choose one of the million, and then do in vitro assay, or maybe 10 out of the million, and then confirm it by in vitro assay. Is it the best way to do? Or do in vitro assay first, see the active compound and the active compound in vitro, or maybe in vivo. But usually we do in vitro, just in the lab, on the cell lines usually, or cell-free on the protein, of course. And then do the docking to confirm the in vitro activity. So in approach, I would do the second one because it will be more appropriate to confirm the activity from in vitro, because we have a limited assays. These assays have limitations because they not ultimately, because you have one very optimal, you give antibiotics maybe, very optimal condition, but in the body, there interact many tissues, many factors, microbes, etc. So in vitro, it is already limited, but we may see that there may have potential time and also expensive. So need to always consider what are the possibilities. So in this study with Dr. Eman from Egypt, they isolated one tannin type, allergic tannin type compound. And during our screening, we found it active with IC50 about 2 micromolar for elastase release. And then we also tested the elastase enzymatic assay. In this way, we confirm it's interacting with elastase directly in the vial. So then, okay, why not? We take it for the docking. It should be docking as well. Let's try to do that. And then I will show you the detail of this part, the docking of this allergic tannin. It was, by the way, a new compound, but the elucidation is a little difficult in this case because it has so many signals. And I will show you the detail of this. Also, they did the hepatoprotective activity in a mice model. So in this poster that we showed together, you can see that the bioactivity is concentration dependent. And then we aimed to simplify this molecule, to remove this flavon type, flavon part on the molecule, on the polyphenolic scaffold. And this is representing by this compound. rhoptalainins, that is simplified version of the big tannin, and we docked them together with some of the positive controls as well. And you can see the results. The binding is here, the interactions, the interaction with the hydrogen bonding actually with the protein is marked in green. So these are the amino acids that are responsible for this interaction in the protein structure. So that is basically hydroxy group with some of the keto group, and they between each other, they will have interaction. And then to basically quantify or evaluate the efficiency of these compounds, we can see the binding energy. The lower, the better. So civilestat is known inhibitor of elastase. It really binds well and it really docks well. So everything is fine. Everything is as expected. The civilestat works, positive control works. Our compound is weaker and the smaller molecule fragment is stronger than our compound. So in this study, the results were, I would say, logical. And that is for the auto-docking example, because if you have any question, you can ask me in the end for more details about this. Basically, I use this free system auto-dock because it's available. You need the ChemDraw. You need a database that there is available for free, protein data bank. Then you need this auto-dock software. It has some tips and tricks. Some of the proteins, if it's too complicated, may not work. But always in the experiment, that's like that. And for the second part, ChemGPS. What is ChemGPS? You know GPS, the positioning. Everybody uses it every day, GPS. So the satellite kind of positioning. This is global positioning system in chemistry and is related to nature products. So there is one without nature product, there is one with nature products. And this is developed by Professor Backlund, Andres Backlund from Sweden. And because he brings students to global pharmacy for last 10 years, except of COVID times, last 3 years they were not coming. So this collaboration also, he introduced this method to us. We work together to apply it for our research. And I think not many people know about this gene, especially nowadays the GNPS, it's much more powerful, but it's different. So I don't think that's a problem. It can still be used this way to correlate the activity and the structures. So what we are talking about is the tool for navigation in biologically relevant chemical space. What we need is the smiles that we produce. Okay, PubChem has it, or ChemDraw can produce that. And this software is online, is free of charge. Sometimes it doesn't work because some of the license problem, but most of the time it can be fixed. 80 chemical properties of every structure will be put into 8 dimensions. So the chemical properties are different: polarity, hydroxy, double bond, electronegativity, etc. And we visualize 3 dimensions. And in a very, very, very simplified summary, the red axis is the size of the molecular weight. So the L-arginine that I showed you will be like 1,000, it will be very high. The orange is aromaticity. Again, that polyphenolic compound will have a very high level of aromaticity. So in orange axis, it will be very high. Green is lipophilicity. So if we have less OH group, then it will be very high. For example, sugars will be very low. So I'm coming back to my original topic, which is the fatty acid, because you know fatty acids are nonpolar. So if we position the fatty acids that I worked with, about 30 fatty acids, you can see some difference between, I zoom in here. So the difference between the hydroxylated fatty acids are here. And then there are the more, let's say, common fatty acids, like without double bond. So the hydroxylated are a little bit out of this cluster, is a little bit shifted from this cluster. And then we try to correlate this activity to the clinical drugs. If we position it, these two pictures are basically the same, just move around. This is the 3D. It's hard to show the 3D in 2D, you know. So if you move it to this direction, you will see that the red axis and green axis is actually similar. They just differ by aromaticity because fatty acid has no aromatic ring. But clinical drugs like Zyrtec and even dexamethasone is here because it's used, some steroids. They are on the opposite side due to their aromaticity level is much higher compared to fatty acid. So these clinical drugs here share some of the physicochemical properties regarding the size, molecular size, and also lipophilicity. But they differ in aromaticity. So we can do the correlation of this. And this is quite interesting because if you find some active, actually these were not super active in the allergy. So it's not totally good example, but it's at least to show the approach that we can do. And in another example, Kartiko Arif Purnomo from Indonesia, our PhD student in Professor Chang's lab. So I tested his compounds for anti-allergic activities, and we identify some of them as active. And also the dual antihistamines, because I take one as a positive control, were positioned in very, very close to each other. So we can correlate these active compound with some of the drugs, clinical drugs, that can share the properties, the physicochemical properties. And because I also tested the clinical drug, so then we can see the in vitro result, and we can also discuss about the physicochemical properties based on this prediction, because ChemGPS is still like prediction about the chemical similarity of the compound. Because nowadays, this, for example, our compound may be alkaloid, but the active compound that is used in clinic may be steroid, totally different structure, but in the end, they may share the physicochemical properties. So this is the aim of ChemGPS. You can correlate totally different structure types to each other, and you may find they may have activity in some of the assay that you choose or some of your target bioactivities. So it's not only the same skeleton will have similar activities, but different skeleton can be correlated to each other. And in the third example, this is the ginger compounds that were isolated and then synthesized derivatives from ginger, so including paradol, sugar, and gingerol. And here, this is my classmate, also from the PhD course. Here you can see this huge cloud of compounds put into the chemical space. This is the TRPV1 agonist database. So they can induce the TRPV1 receptor, which is the pain receptor. So this is the receptor for pain. And we know that capsaicin, which is derivative of gingerol, also its structure is similar like this. Capsaicin is also TRPV1 agonist. So then we correlate the ginger compounds if they may actually work with the pain in the same way like this pungent capsaicin, like a spice in chili, right? So these derivatives, and you can see them nicely together, with capsaicin. And also some of the inhibitors are there, but actually there is a gap between the clinical and the official TRPV1 agonist and our experimental ones. So this is another interesting example how to compare activity for the series of compounds. And okay, in this example, we recently worked with Ukraine to correlate the phenolic compounds isolated from iris on the H1N1 influenza. So these were active, the red one were active, and the drugs for the virus treatment. So we can see some of the similarities with this yellow cloud. And orange is, for example, the neuraminidase inhibitors, which is then the specific pathway of the mechanism of action. Okay, I wanted to show you example, how does it work here? I hope you can see it. Basically, we use this software on Mac, and these are the compounds that you position in the system, and then you can control which one you will show up because you already prepare the different groups, and then you can basically take a close look on any kind of certain point. You can modify the colors, and you can try to think like what these two are and what kind of correlation they may have, what activity they may have. So this is just to demonstrate how it works. And the last part is the global network positioning. So like global nature product social molecular networking. Yes, that is the abbreviation GNPS standing for. So GNPS is different. It's based on LC-MS. So you need to do HPLC, high pressure liquid chromatography, usually it's ultra high, and then submit to mass analysis. So I will just introduce the basic approach. And some of my collaborators trying to observe it in the moment. So in this study, the authors use the extracts, 2000 extracts, to find some of the active ones, active extracts. So they use bioactivity. Plus, they do the HPLC and mass. So they can see the structure fragments. And based on the bioactivity and these fragments, they can choose, it's so-called structure-based annotation, they can choose some of the network that is done by the system to discover the new drug or discover the active compound. So they need to organize the data, they need to link the activity and the structure to target their compounds that they will in the end isolate. So this is one of the interesting approaches that was designed by the other researchers. Because this is like, and then you can prove that this prediction was correct. So how does it work? You collect the LC-MS data, then you server submit to GNPS, and then you have molecular network. MN is molecular network. So molecular network looks like this. You will get something like this, cloud of huge amount of dots. The problem is how to get oriented in this and what kind of compounds are there. So because this is usually untargeted, so we don't know what is it. We don't know which compounds there are. But the database online can predict based on the high resolution mass and formula and the fragmentation. It can predict which compound it is. So in this paper, they use the bioactivity fraction, basically bioactivity fraction, to limit the amount of the dots to the certain amount only. And then in that final chosen already like reduced network, we call it network or cluster, they were able to predict these structures and in the end confirm by isolation this and this compound. So they went back to big batch and they do extraction isolation and they try to get this type of compound. In the end, they proved that there is this compound. And it's also related to bioactivity. So this is very interesting approach, but usually it is hard to get such a ideal situation. Why? Because when you have many, many compounds in your sample and you get the mass profile, thousands of mass profiles, because it as well as 1 gram of compound in your sample, and it will detect it equally as one spectrum. So then you don't know which one is the major and which one is the minor. The only way to know is to see the UPLC profile or have a standard compound. And then basically the mass data for you. So it will make it into clusters. For example, if this compound has some long chain, maybe this one has some long chain, 90 and 147. Okay, so both have 57, both have 90 fragment, and both have 147 fragment. So they will be linked in your cluster, in your network, through that certain fragment that they are sharing. So you can see like these are sharing the fragment, but these are not sharing the fragments. So these will be discarded. So they will not be part of this cloud. They will be in the end separated as single dot in the end. That is basically non-networked, non-correlated one. Yeah, so this is the GN, and now because also the timing, I'm not so sure how long I take. Here I will do some of the introduction of our basic work to have the target-based extraction of bioactive components. Although it may not always work, but at least we try our best to have some specific target, for example, bioactive or specific structure. And also bioactivity guided fractionation. We are trying to collaborate with the biological labs to screen bioactivity and work on the active fractions. And then we apply bioinformatics to prove these. And I want to introduce, if anybody is interested by the research that we show here, any part of it, folk medicines or bioactivity guided or isolation, chemistry, bioinformatics. We have in Taiwan, so-called Taiwan Experience Education Program. This program, so-called TEEP, T-E-E-P, TEEP. If you, right now, if you have a phone, you can take this QR code and you will get to the web page of TEEP. And the TEEP program, maybe I could show you how does it work. If you want to search, if anyone is interested, there is no any limitations for your degree. You can be bachelor or master or PhD. Basically, I just type TEEP and I'm there. You press find the program, and here you can put public health if you are interested. And then if you click search, it are often pop up and select which one you want to do. So you can see we had 2 hits. Okay, I type nature product. So if you want to work with natural products, you may see the 3 of us is from our university. So this is Professor Huang that do the neutrophil study with us actually. So if you click discovery of them are also from our institute. So it's Professor Yen and Wang. Here is basically and get if you're not discount, we can get in contact. So if anybody is interested, please welcome to join us. What is the condition here? So like I say, no limit for the people's education level. I think the most important is to be interested, to explore. And it is good to have basics in chemistry if you join our chemical lab. If you want to do biological lab, pharmacological professor can also accept, we can collaborate, we can figure it out. So why do you want to join? Maybe you want to study your favorite plant or food extract or your grandma's treatment, or somewhere in the forest you find something and then you are interested. And how to survive in Taiwan for this stay. Because this program supports 15,000 NTD per month scholarship and we have 6 months, but you can also join for 3 months, it's fine. Or summer vacation, okay, no problem. 2 months, 3 months, fine. So if anybody is interested, please go ahead and contact us here. And also, like later, I will ask Vidya to introduce also. We also have a master and PhD program in our university, not only our natural product department, but we have also clinical pharmacy. And we have all different technology and so on. So fragrance cosmetic department also is a big department for fragrance products. So all of the program, but fragrance maybe not. Some slight differences, but anyway, if anybody is interested for master or PhD degree, also very welcome. And it is for masters, you need to be graduated as a bachelor. Basically, that's the requirement. For PhD program, is recommended to be a lecturer because then you can get very nice support. You can also join without being a lecturer like Eco did, but then the support we cannot apply like that. So that will be a much limited funding basically. But again, if you are interested to explore and brave to come, very welcome to join. Yes. So here I mentioned find us and also our department, natural product chemistry, Professor Zhang Xinsuo is affiliated from the Farmasi College, also doing the chemistry, and myself, Professor Zhang is focused on epigenetic from fungi, but also do the plant material, and Professor Xinsuo is focusing on Formosan plants, and he make a database of Formosan plants. And Professor Wang on cancer biology, Professor Wu on antiplatelet, Professor Yan high throughput drug screening, Professor chain, on bioimaging. So all these 4 professors are very excellent in pharmacology. That makes the pharmacology part also very strong in our institute. So if you come to our lab, basically this is how it looks, to have idea for you what we are basically doing, or what we are focusing on. So if we have plant material, we will do the extraction, ethanol, methanol, partition, EA water, or acid-base extraction for alkaloids, for example. Then, of course, TLC is necessary to do. Sometimes some interesting shapes are coming. The column chromatography, separate compounds, precipitation, separation, then structure elucidation. These are the important parts. We also use MPLCs, HPLC, and we can separate the compounds for some analytical type of columns. Very good example would be like this, but it's usually more broad. And we also have rotation level operators, centrifugal level operators, NMR we have in the university. So we are able to test NMR anytime that we can do NMR-based isolation. I think this is the signals like I showed in the previous study. And here I just want to briefly show who are currently in my lab. So Yenchi is now the 3rd-year PhD student who is already trying to finalize her study. And she is from Malaysia and originally graduated in nutrition, able. That is like a food from Malaysia. Agarwood is currently he's in the PhD program actually. He directly changed to PhD program. So Tiago is from India and he graduated as pharmacist in India, and he is studying composition of different colored flowers from Leonorus japonicus, and now also a special food vegetable with chicken. The leaves are added to the chicken. We are now studying that because it's seems to be good folk medicine leaf. So that is another topic. Guanghan is studying the compounds from Taiwan endemic plant. And Sunny, he is a visiting student from another university, and he studies the compounds from the sea corals. That is very interesting, with anti-cancer potential, but these are very few. The amount is limited. That is a big limitation for this kind of study. And also we have Vidya, that I don't have a slide here. I don't know if Vidya can hear me and can introduce briefly herself and her study.
Yes, Professor.
If Vidya is available. Okay, you can also speak in Indonesian. It is very okay.
Yeah, okay, thank you. Yes. Oke, halo semuanya. Perkenalkan, saya Vidya Pebraska Tenderli. Saya dari lulusan S1 Farmasi Bahan tahun 2016, lulus tahun 2020. Sekarang saya sedang melanjutkan studi master di Kuliah Medical University, lebih tepatnya di Graduate Institute of Natural Product. Kebetulan di labnya Profesor Michal, dan sekarang saya sedang meneliti terkait salah satu tanaman untuk isolasi alkaloid, dan selanjutnya untuk untuk antivirus. Mungkin kalau misalnya ada pertanyaan atau misalnya ada yang tertarik terkait internship di Taiwan, bisa hubungi saya. Sangat senang sekali kalau misalkan ada yang tertarik, and please welcome to our lab. Yes, it's very nice to be guided by Professor Michael. He's really nice teacher. Thank you, Professor.
Oh, thank you so much. Thank you so much. Okay, so just to maybe summarize in 5 more minutes, and then I will just have some photos. So what we are doing, or what is our thinking, or what is our aim, why we want to do this kind of research or this kind of work? Because if you are now in master, okay, as a pharmacist, we have in Czech we have directly master, so I always think it's master directly. If you are in your bachelor degree, for example, you may think, okay, I will go to pharmacy to work or industry, or can I maybe think about some new ways to explore, to do research, to understand the folk medicines, to classify, prove scientifically some of my grandmother's favorite treatment. Was it done before? It was to explore. Where is it? Can I find it? Is it a waste product from the industry or from people that they use to utilize this kind of material? So we ask a lot of questions, basically. And then because we are now working in the chemical lab, but we collaborate with the biologists. So what are the chemical compounds? Crude extract activity, and we want to make the correlation with the, like, clinical drugs are chemicals, right? So because they are pure chemicals, pure chemistry may have toxic effects, may have undesired effects. In mixture, the plant may have less of the active compound, but synergistically being less toxic and then being actually more useful. So that's why we also need to understand the chemical compounds. It is because the Western medicine is only friendly to the Western pure compound drugs. But nowadays, it is changing back to nature products being very, very important. And you know that nature products are sources for these synthetic drugs. Maybe more than 70% is either direct or indirect derivative of nature products, like papaverine, morphine, nicotine, caffeine, codeine, etc. So everything is from nature. So it's coming back, and we try to explore these. We try to ask a lot of questions, but then in the end, we need to understand the chemical part of it. So this is first our questions, and then is a lot of lab work, and also to explore the unknown compound in the sample is another challenge. And sorry, I have some. Okay. Computer got a little froze. So, like I said, so with coronavirus, also many people came back to nature product and related research. And we also collaborate with Professor Huang, who is doing essay with us for this activity, and with our other labs also we collaborate. We are a little mixed up with the other students from other labs. So some of them are doing chemistry as well as bio. We have also other members in the lab that are not directly inside. And also, of course, Professor John Van Room's lab has a lot of students. So this is what we are doing: extraction, isolation, structural elucidation, HPLC profiling, and bioactivity evaluation. And one of the example for success of Taiwanese research on herbal drug is Rispar 8. This is approved in Europe. This is approved worldwide. It's based on TCM. It's approved by many in vitro in vivo assays. And this is one of the examples where we can apply our skills that we learn in the lab. So we also need to logical thinking about the problem, which sample we will work on. And of course, work and communication. So the lab, and Pingwei was working on the chemical part, and he will come back to join PhD, Taiwanese. And so who can join us? Anybody, like I say, who is curious, or just choose the TEEP exchange program is fine. For lectures, maybe we can also figure out a way to have the collaboration or the short exchanges. And this is my acknowledgement, and then I will have just some photos. So of course, many people in Taiwan, in Professor Chang's lab, Professor Wu, now he moved to Taichung, Professor Chen Binghong's small lab doing anti-allergy assay. So I very appreciate that. Then the other Taiwanese and International Mohammed Al-Shazli, a big support from Egypt with his groups of collaborators. Joe Stoker, American, who is on folk medicine, and he studies like cookies in the lab and so on. So it's like cultural way of thinking. So that's also very nice. Atolga Michal, big support from Ukraine, and we support so much Ukraine from Czech is pharmacognosy members in Charles University, Faculty of Pharmacy that we have. Actually, in Czech, there are only 2 faculties of pharmacy in the whole country. Ending up on this session. So we have many online meetings. We tried to make up international project with Ukraine, with Egypt, Dr. Shazli, and the faculty members in Faculty of Pharmacy of Shams University in Cairo, in Egypt, also pharmacognosy. So all of them are pharmacognomists. Professor Huang is then a pharmacologist for the inflammation. Yes. So in Taiwan, like this is the Synapse classroom. This is, you can see many food that hot pot or I think Indonesian like food a lot. Taiwanese food is a little bit different, but definitely good to explore. And we have the activities like this. We go with the lab, Kumar, that is professor from India. Yeah, members that are international as well. We went to sacrifice something for the gods. This is a tradition for the lab, basically, every year. Yeah, so here, okay, Vidya is here. So when Vidya come with the other group of PhD or master students, so we went out together to, and here's Tiago, and this is my family, and this is a bridge in Kenting, and my 4 years old Yano and Michal is 10 years old. So yeah, some point to like Czech event in Pingtung. So you can see on the map, interesting to see some Czech activities in Taiwan. So we also like to join it. Okay, so terima kasih banyak. Thank you so much.
Well,
And if you have any questions, then you're very welcome.
Okay, thank you very much, Mr. Michal, for a very interesting lecture. I think you've done many research on natural product isolation, including the activity assay also, and then the bioinformatics, as you said, to confirm the in vitro activity assay. Okay, I see there is someone raised hand on our Zoom room. Saidin, do you have any question for Mr. Michal? Please.
Check. Am I audible?
Yeah.
Okay. I have some question for Mr. Michael, but first let me introduce myself. My name is Edin, and I am currently studying in Universitas University, and right now undergoing my research in bioinformatic field. So, Mr. Michael, I'm interested with your explanation about GPS and P that used for navigation. So, if I want to combine GPS and P with molecular docking in one research, which one should I do first? Is it molecular docking or is it GPS and P? And also can this GPS get toxicity data from the chemical that we use in the GPS and P? I think that's all from my question. Thank you.
Thank you so much for your question. If you ask about the GPS and P and the toxicity data, from GPS and P, we can only know the compounds data that you get from the database, or you form the database. For example, you go to ChemBL online and you search all the approved drugs. Actually, you make a term like toxic to this kind of cell line, for example, or you will put certain molecular target, like inhibitors of this pathway, like apoptosis related markers, and then you get all of the compounds that are reported, that are in ChemBL, that are in this database, and then you will input them. You will export them as smiles, and you input them into ChemGPS, and that way you have this kind of like a group of compounds that are toxic to compare with your compound. But of course, you cannot, unless it's reported, you cannot know that your unknown compound has certain activity without doing the real bioassay. If you want to know your compound is active, you need to do bioassay, or you need to check that somebody did it already and published on apigenin being toxic to the hepatic cells, for example. So what we do is that we do the bioassay and first, and then we position it into ChemGPS, and then we have some control group of compounds that have activity. We know them, that they are active. They may be clinical drugs. That is the best way to do clinical drugs, to put into this kind of small database. Some databases are available. Professor Andres Backlund may have some of the specific databases. He collaborates with many of the companies. So online there are available, or via collaborators we can obtain some of the databases. But like I say, if your compound is toxic or not this is only prediction. So this is only correlation between some of data, some data that you input and you know it is active. You can compare and you can see the dots are close to each other or not. And this is the only comparison we are doing. We just certain physical chemical properties, attributes, features, which they share or they don't share. And then we assume, we make like prediction, they are active or not. So the ChemGPS is physical chemical properties prediction, similarities prediction. So that's, and it's related to nature product. So this is adjusted to fit more nature product than the synthetic, but the synthetic is also possible to put in, of course. Yes. So I don't know if this is fine for you to answer. For molecular docking, which one to do first, ChemGPS or molecular docking? For this question, I think it is case by case, because in my opinion, always the in vitro assay should be first. But you can see many researchers only do the bioinformatic way. And I don't know if you are bioinformatic researcher, if you have collaborators who will do any kind of bioassay for you, or you will take reported active compounds and apply it in the bioinformatic to prove certain activity. So that is also possible. Yeah, that is also possible. Or just like I say, the first version, like you only predict, you only do the docking and see which one has the lowest energy, and then you propose that this is the promising compound for the future development. So this is also possible, but I would like to say the limitation of molecular docking and also ChemGPS is they only work with pure compound. If we have a crude extract, there are thousands of compounds inside, we cannot understand how they work together. So these both systems can only work with the pure compounds. Sometimes you can dock two compounds into one molecule, into one protein, but I think these are then so complicated softwares to do that. So I don't have so much experience with that. So we only do one compound at a time, always. So I think this is the limitation. Yes, thank you very much for your question.
Okay, thank you so much for the answer. It really
Thank you, thank you.
Thank you.
Thank you.
Okay, thank you. Gimana Yedin? Sedin? Cukup.
Alhamdulillah Ibu cukup udah terjawab pertanyaan.
Okay. Anybody else who want to ask a question for Mr. Michal? Boleh dalam bahasa Indonesia, tidak apa-apa.
I think you can also type the question.
Yeah.
So far, I didn't see if I can see well. You can, don't worry, just type anything. Like, for example, why did you come to Taiwan or anything? Any question is welcome.
Ya, mungkin tidak harus pertanyaan terkait materi juga katanya, boleh apa namanya, mungkin opportunity untuk studi di sana, boleh ditanyakan.
Or Vidya, do you have any question?
I think somebody has question.
Somebody ask a question, have a question
On chat box.
column chat from Reprianti Irma. I will read it for you maybe. Hello, Mr. Michal. I want to ask about GNPS. Before that, let me introduce myself. I am Refrianti Irma from Buana Perjuangan University, Karawang. The question is, why did you use GNPS to analyze the data? Why don't you use other tools that match the data you want to know? Thank you.
Okay, thank you. Thank you very much for your question, Refrianti. Why did you use GNPS to analyze data? Okay, the example I chose is taken from the reference. It's not our study, to make it clear. Like, for example, our classmates, they use GNPS, but from the crude sample, but they already isolated during the master course. One of our classmates isolated about 20 compounds, coumarin type, and then they sent the crude extract to GNPS analysis. And because these pure compounds were known compounds, and they also did some bioactivity in the meantime. So why do they want to use GNPS to analyze the data? This is the main reason is to understand the composition, not only the compound that you isolate, because the minor compounds we will not isolate, the compounds that we are not interested, maybe some fatty or oil, or we will not detect it. Some compounds are maybe easy to decompose during the column chromatography, or the column, the silica gel. You will not detect it. And then the LC mass will give you very detailed analysis, prediction on the structural features that you can then predict by using online databases. And if you have your standard compounds, like in our case, my classmate, then we can make sure that that compound, because the LC retention time will be the same, the mass and high resolution mass we submit from saturation, etc. So you have many information from the GNPS system, especially the high resolution one, that can prove the prediction. Like, I don't say 100%, but maybe you can almost get 95% correctness prediction. But in the GNPS, because we work with high resolution mass, the ultra high LC, which retention time will be so sensitive. So then we can, and we work with very few amount of sample. So these are all advantages to understand complete and unchanged and the real composition of your sample. But again, the limitation is with the prediction. It's just prediction. If we have standard, we can maybe prove it almost 99% that this is the researchers use. And maybe Mr. Sedin will also want to use this tool in the future to match the data, to match the basically match the composition, match our crude extract composition with the real composition. So although there are limitations, but it is very useful tools and possibilities. So in the future, I think we also need to learn more about this. So we don't want just to stay in the wet lab, but we also want to use some of these useful tools. So of course, it depends on the maybe on the topic, on the research ideas, on the literature, that was done before, always we need to check what research was done. If somebody did this already before, if there are many compounds isolated from the plant, and so on, everything we need to consider. Here docking, if you have bioactivity with enzymes, it's easy to dock with. Another bioinformatic tools, there are some engines that can do different things between chemicals and proteins. So, or NMR prediction is there, right? There is, based on NMR comparison, the extract can be also compared. There are different engines. So how we choose the tool only depends on our study design and also literature background. And if we have the compounds standards or we don't have, is it pure prediction? So it's complicated to have clear answer for your question, but we need to consider about all of the background, all the data, and what is our aim also. Yes, thank you.
Okay, thank you. So I think both GNPS or chain GPS was a tool to, what is that, to find the correlation of the activity from a compound based on their similarity.
That's
right, from
correct.
chemical properties, or was it spectrometry data, like that, I think.
The Chem GPS is based on correlation between physicochemical properties, like you say, relate to the bioactivity. Yes. For GNPS, the bioactivity part is a useful addition, but it is not necessary. You just want to analyze the chemistry of your extract. The study I shown is example where it's correlating the bioactivity inside. Okay. That's why I like it, and it's an interesting approach. Yeah, but it's also not very easy to fulfill such a study, because there are many
that we use is depend on what the data we have. Yeah.
Yes, because in that study that I showed, the authors did bioactivity first, and then they reduce the amount of chemicals. They reduce the amount of chemicals based on the bioactivity active compound.
Okay.
Yes, so Chem GPS with bioactivity, GNPS, LC mass is more on the chemistry. I identify the component. But we don't know how much is each compound. That is the limitation. It may be 0.1 milligram in all plant. It's still 1 dot. It may be 1 gram major compound or 20 gram major compound in the extract. It's still 1 dot. We don't know the amount difference in the network. Yeah.
Okay. Any other question, maybe?
Yes, you are welcome. And also recently we focus on one of the bioactivities from the wild plants that contain alkaloids that is active for the coronavirus. So it is for berberis type of plant. So if you have in Indonesia some very popular species, it may be interesting to maybe collaboration or consider.
Yeah, of course, because like what's that? Typhonium or aqualira Aquilaria, right? We have also the same species here in Indonesia.
Mhm.
So I think,
Oh, okay, okay.
yeah, I think there are many natural many plant here in Indonesia that have possibilities to be further research for its activity. Okay, I think we don't have any other participants who have questions. So before we end the session, I think we need to take the photographs together. So to all the participants, can you please open your camera? Bu Mira bisa dibantu?
Ya, baik. Di sini ada 11 slide. Slide yang pertama, mohon siap. 1, 2, 3. Kemudian slide yang kedua. Slide yang ketiga. Slide yang keempat. Slide yang kelima. Slide 6. Slide 7. Slide 8. Slide 9. Slide 10. Slide 11. Ya, terima kasih sudah dipoto semuanya.
Terima kasih.
Okay, and for the last semester, we have a virtual certificate here that we will send you. I will share it on the screen.
Okay, thank you very much.
So here's the certificate for Pharm.Dr. Michal Korinek, Ph.D. as the guest lecturer on the theme of bioactive natural product explored with the aid of bioinformatic tools, held on Saturday, February 25, 2023. And I think we can end the session, Mr. Thank you very much for sharing us the knowledge, for willing to spend your weekend time here with us. And happy weekend, I think. Have a nice weekend for you and your family.
Yes, thank you very much. Yeah, I closed myself here, so I'm not disturbed because the kids are already noisy there. Yeah, thank you very much, Dr.
Yeah, you're
Tresna, for
welcome.
your kind invitation and
Okay,
all
I
of
think
the related matter with the lecture. Thank you so much. Thank you so much. And everybody is welcome to contact me if you are interested to come to Taiwan, or if you have trip to Kaohsiung Medical University, we'll be very happy to meet in person. Thank you.
Okay, thank you very much, Mr. So I can stop the meeting now, maybe. Ya, sudah bisa meninggalkan room Zoom-nya ya. Terima kasih semua untuk yang sudah hadir. Mudah-mudahan ilmu yang diberikan bermanfaat. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Terima kasih Bu Tresna. Thank you,
Ya,
Mike.
sama-sama.
Thank you, Mr. Michael.
Mister.
Oh my God. Kita lihat ya. Oke.
Recording stopped.